Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this is number 65. And it's going live on June the 26th, and it's even warm here in Scotland for a change. It's also perfect for sports lovers, as we have the World Cup, Wimbledon, the Tour de France and baseball all at the same time as of next week. Last week our guest was Bora Pharmaceuticals Chief Commercial Officer Jean-Baptiste, also known as JB Agnes. This week it's a conversation with the CEO and co-founder of a newly launched company, Houdini Bio, and that is Jonathan Cohen-Gold. If you do have news or suggestions for who should be on the podcast, or indeed any other comments or suggestions, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us, or sponsoring a podcast episode, or for event coverage. But first, it's time to go through some of the headlines of the articles you may have missed over at deeptechdigest.com over the past week. Speaking of Houdini Bio launched to solve therapeutic and commercial genetic medicine bottlenecks. Kither Biotech completed its phase 1 trial of an inhaled peptide therapy for respiratory diseases, and In Silico Medicine and SK Biopharmaceuticals are set to collaborate after signing a $2.5 billion deal. Lab-engineered proteins could be a promising treatment for liver disease. Could food influence gene activity, we ask? And Meteor Biotech joined a brain organoids consortium. Memento Medicines launched with $93 million to develop an antibody therapy for retinal diseases, and Vedana Therapeutics launched to develop migraine therapies. And you can read all of these and more at deeptechdigest.com. All right, let's get to this week's interview, and this week it is a conversation with the CEO and co-founder of a newly launched company, Houdini Bio. That CEO is Jonathan Cohen-Gold. We'll find out why Houdini, we'll find out about the Hush complex, which isn't about being told to be quiet in a movie theatre, and we'll find out about more affordable, durable gene therapies and cell therapies. All right, hi Jonathan, great to have you on the podcast. I wonder if you could fill us in with a bit of the story so far leading to the company emerging from stealth recently.
Yeah, absolutely. So it all started when I began my PhD at Cambridge. So I was joining a group run by a guy called Professor Paul Lehner, and he was a virologist at the time. That was his major focus, particularly on HIV research. And he discovered this about 10 years ago, so 2015. A completely novel group of proteins that was called the Hush complex. And he described it for the first time, and it was a kind of cellular machine that defends our genome from viruses, and particularly from HIV. So he identified Hush as really primarily responsible for identifying HIV inside the cell, inside T cells, and silencing it epigenetically. So that's where it began, but the super interesting thing actually happened after that when I joined the lab, in my opinion, when we realized it wasn't just HIV that Hush was silencing. It was actually any piece of foreign DNA we put into pretty much any cell. And that includes therapeutic DNA as well. So a bit of a light bulb moment happened, which is, are we battling against this in the clinic as well? Are some of the challenges which we're going to go into in a bit a result of Hush turning off therapeutic DNA inside patients? So I then spent my time at Cambridge really focusing on escape routes. So really kind of deciphering the molecular determinants of what Hush recognizes as self or non-self. and using those rules to try and engineer an escape route around Hush. And so I found some really interesting things. I embargoed everything because I knew I wanted to return to this at some point, but I didn't really have any idea. I didn't really know what venture capital was at the time. That was a completely new concept, and I knew I needed to learn about it. And I was able to join a VC company called Deep Science Ventures in London. My boss there, Kirsten Pappenfuss, she was brilliant and just said, come and work for us for a bit and get an idea of the lay of the land and how things work. So I went there, and then eventually, when I was more familiar with that kind of world, decided to return to the science. I met my co-founder, Lee Dunham, who was the commercial director at a big cell and gene therapy company in the UK. And we both built conviction in the idea, decided to quit our jobs completely, and then start Houdini. So that's kind of the origin story of it.
Where did the name come from?
So yeah, as I kind of alluded to it in the answer, because I was really obsessed with this concept of hush escape. And I kept thinking, how do we escape this thing? And for some reason, my mind then went to an escape artist, Houdini, and I thought it would be memorable. What I found at the beginning is that some people really, really loved it, and some people really hated it, but everybody remembered it. So I was happy with that. And then we've kind of just gone fully into Houdini and that concept since, and that's how the name came about.
What was the gap in the industry that you saw that made you want to go in this direction?
Yeah, I would say I probably wouldn't describe it as a gap. I would describe it as a chasm. I think what I saw was, and what my co-founder saw on the industry side as well, was that on the one hand, you have these curative medicines that can treat disease we never thought was possible to treat. We have some proof points in the clinic of people being able to see for the first time, or being cured of muscular dystrophies, for example, and being able to walk for the first time. And so the proof points exist, but in the middle is a huge market adoption problem. So these gene therapies often cost about $1 million a dose to make. And as a result of that, they're often priced $2, $3, $4 million per patient, which is a really, really big ask. And if you're going to ask for that much, the therapy better work for the entirety of that patient's life from a healthcare economics perspective. But otherwise, the whole payment structure and value falls apart. And that's what's been happening. And so we've seen that on one side, we have these incredible medicines. On the other side, we have patients that want them and need them. The science is there, but there's this hole in the middle, this chasm in the middle. And so we think that it is an addressable bottleneck. The main kind of take-home message is we shouldn't be giving up on gene therapies. We should really be trying to solve what we think is a solvable bottleneck in the middle that's currently disconnecting those patients from these amazing drugs.
And so, what is that bottleneck?
Yeah, it boils down to the fact that therapeutic DNA is remarkably inefficient inside cells. So back to the Hush complex that I mentioned at the beginning, therapeutic DNA, it looks nothing like our own genes. And so the body recognizes it as an intruder. It looks far more like a virus than one of our own genes architecturally in terms of its sequence compositions. So the cell shuts most of it down. And so what you end up with is, firstly, that explains the kind of fading efficacy problem that we see with gene therapies. Sometimes this can happen during a clinical trial period itself, and then endpoints aren't met and the drug is not carried on. Or sometimes it can happen post FDA approval in treated patients, where you can see that there was an initial effect, but over time, durability wanes and patients can relapse back into disease. And that is a silencing problem. That's DNA being turned off over time in the patient's body by the Hush complex. And the other thing that kind of compounds this is, if only a fraction of the DNA is working, you have to dose incredibly high to get therapeutic benefit. And so what we're seeing with AAV therapies, for example, is we're dosing 1,000 trillion DNA copies per patient. And to produce that much material costs a lot of money. The manufacturing process isn't very efficient to begin with. And so it's really a case of the huge doses required because of this also brings up that price. It can also be really unsafe. We've seen patients die from this kind of dose-dependent toxicity. So really, we think that DNA silencing is underpinning this core set of challenges that's currently just trapping the potential of genetic medicines.
So how did you bridge that chasm, I guess?
Yeah, so our technology is really trying to make that therapeutic DNA look more like self and less like non-self. So the mechanisms and rules that I discovered during my PhD really formed the basis of redesigning DNA. So maintaining the protein coding function and the therapeutic function, but just modulating the architecture and the sequence composition and other higher order features of a therapeutic piece of DNA so that it's not recognized by the Hush complex. We kind of call it this molecular camouflage. So it can go into the cell, but evade that silencing process. And that means lower doses, lower cost, safer treatments. But also the big win is that these therapies, once they work, our vision is that they'll work for the lifetime of a patient rather than work initially and then eventually be turned off. The real way forward here is encoding resistance into the DNA itself so that just that therapy can evade the system, but its physiological function remains intact.
Is this something that other companies have tried, or are you in your own territory here?
It's a bit of both. So there is space in this field because the unmet need is huge. And I often say that part of the traction that we've been getting so far is the fact that it's on the problem side. You need a good technology, but it also needs to solve a really, really important problem. And so because this problem is so huge, there are companies working in the space. They've kind of been shooting in the dark though. And so our unfair advantage here is that we understand Hush, we understand Hush biology, and also these proprietary ways of escaping Hush. And so instead of taking shots in the dark, we can use first principles biology. We do combine that with machine learning and AI, which, even though it sounds like a bit of a buzzword or something shoehorned in, actually took us much further than we could go without it. But essentially, the understanding of Hush and those rules which determine its escape, which allow DNA to escape Hush, that's our unfair advantage. What I would say is that our technology doesn't necessarily need to replace the existing tech stack, because what we've seen so far is that we can also be additive and in some cases synergistic with existing approaches. So it's really about kind of raising the ceiling of state of the art rather than simply replacing everything. So in some cases, it's good for us to work with current approaches as well and plug into that.
Without giving any technical details away, what's the process that you're using to get this into the cells and evading Hush?
It's a design play, really. So what we do is we have a set of governing design principles, and then we're able to test enormous combinations of these within a sequence space and a solution space that we know is likely to enable Hush resistance. And then we're able to kind of iterate through machine learning models and wet lab validation. And we have a kind of closed loop between those two things. So we can iterate between our designs and their effect, and then predictive models, and then going back and cycling through that. And then the way we deliver it is entirely up to either the partner we're working with, or it's dependent on the payload itself. What we see is that we can have these effects across different delivery systems. So we've seen really encouraging effects in AAV, lentivirus, transposon systems. It doesn't matter, transient plasmid systems. It doesn't matter how you deliver it. Our technology, once the DNA is in the cell, will enable greater expression for longer. And so that's really down to what the asset would actually look like for the company that we're helping develop it with.
You already mentioned the fact that this is an expensive route that companies are taking with gene therapies, but are you envisaging that you'll be able to help with saving money and saving process length?
I think whilst it's about saving, we can definitely enable companies to save money on existing pipelines through using lower doses. So, for example, if your DNA works an order of magnitude more efficiently inside the cell, theoretically, and it's not completely linear, but it allows you to significantly reduce the dose that you're giving and then divide a batch into multiple patients, which reduces the cost per patient and flips the economics. So while we think that's a really exciting value proposition for existing pipelines, it's also less about saving and actually just unlocking drugs that are just tied up at the moment, completely chained up and frozen out because they can't work at all. They would lose efficacy over time, or they'd be so expensive that there's no way the commercial viability could ever be achieved. So whilst we're looking at helping companies save money on their existing pipelines, it's really about unlocking the multi-billions of dollars that's trapped in gene therapies, which would never see the light of day in the first place. So really looking at future pipelines. So that's the proposition from a commercial standpoint, but it's really a win-win because that means patient access, and that means that more patients can be treated with genetic medicines.
What areas of disease or what kind of conditions is this going to be applicable to?
I would split it into two groups. So one of these are your kind of classic genetic diseases, so inherited diseases. Congenital hearing loss, we might think about cystic fibrosis, muscular dystrophies, haemophilia, those types of inherited diseases where a single gene component is driving that disease. But also, a disease doesn't have to be genetic or monogenic to be treated with gene therapy. So we've seen, for example, with cancer, CAR T cells, the cells themselves, obviously, we wouldn't be changing, but they express a piece of DNA to express their CAR receptor, which is responsible for the therapeutic activity of these cells in patients' bodies. And we've seen can have dramatic effects in terms of curing cancer and bringing patients into remission. So we think we can have impact across that space, so across different cancers as well. Also things like neurodegen, Alzheimer's, Parkinson's. This is a very active space for gene therapy. And it's really going to take flipping the economics of it and really ensuring that these do work for the entirety of that patient's life. But if we can do that, it opens up this larger therapeutic space as well, beyond just inherited conditions.
What are your hopes for patients, or what do you anticipate is going to be the benefit for patients at the end of this?
Access. Obviously, it's a huge commercial loss not being able to really capture the value of what gene therapies can do and genetic medicines in general. But, the real human loss is on the patient side because we know mechanistically that these therapies can cure diseases, could completely transform somebody's life or the life of somebody they love and care about. But because of the current bottlenecks, which is basically a technical bottleneck, these patients can't access the treatment. And so really the big win is patient access here and being able to receive the treatment that they need.
One of the problems that there has been traditionally with some treatments, and I'm thinking about things like new antibiotics, there's been a tendency for companies to not want to go there because the profit margins are less. Is there a danger here that companies may not want to do this if their profit margins are affected? I know it's going to be beneficial,
Mm.
but is there a danger of that?
Yeah, it's a really interesting question. It's all part of the same equation. So there's a few ways of looking at it. Well, two main ways in my head. One, at the moment, the market adoption problem, it's not a small problem. It's really, you see pharma companies taking drugs off the market completely. So we've seen even this year, we saw Roctavian, which is a factor VIII gene therapy that got FDA approval, developed by BioMarin, was projected at some stages to generate peak revenue of $1 billion plus. They didn't generate anything like that money and pulled the drug completely off the market this year because nobody would pay for it. And so if you look at it from that angle, it's really a case of not being able to capture any value, or perhaps you can be more competitive in terms of pricing, but you're going to be able to treat many more patients and to generate much more revenue that way. What I do think, though, is that in some respects, it might be a red herring because although $3-4 million, it sounds crazy to be charging that for a single dose of a therapy. When you do the healthcare economics, if that therapy does work forever for the lifetime of that patient, it actually makes sense. It's actually cheaper. So it's not necessarily about reducing price per se. It would allow pharma companies to be more agile with their pricings. They could be more competitive. But really, if you can ensure that a gene therapy works forever, the healthcare economics don't look so bad, and it might not be a case of having to sacrifice revenue in that way.
Well, and as you said as well, that by making something work, you're making the difference or making a choice of something that goes to market and something that fails. So that's a huge difference.
Exactly.
Mm.
Exactly.
So, what are the next steps getting this out and partnering with companies, or are you already in partnerships?
Yeah, so the next step, zooming out, is to bring in some more money. So we're opening our seed round this month. We've been able to bring in around 1.5 million in pre-seed and non-dilutive grant funding so far. And that's enabled us to hit some really key milestones ahead of time. And what we now need to do is expand the team so that we can move faster. And so we'll be opening up that seed raise basically now. What we want to do is, on one hand, we really want to invest in the platform. So our AI wet lab closed loop system discovery engine that I talked about before, we're seeing some really exciting things from that. For example, we're seeing second generation versions of our modifications come out through that model, which are already out competing the first within 6 months. So we think we're scratching the surface and we really want to push that harder. But also, a lot of the work that we've done at the moment has been with primary cells and other cells, but it's been in the dish, and we're really moving forward to more in vivo work. So ideally through partnerships as well and collaborations, we want to start actually taking real world assets that are going through live pipelines and really put them through different in vivo disease models, validate the technology that way, and do that, as I said, through commercial partnerships and collaborations where possible. So that's kind of the next steps for us as a company.
How are you looking to forge those partnerships?
We have been active for a little while, but particularly now we've come out of stealth and we're shouting about what we're doing. The answer really is my co-founder, Lee. So Lee is really the interface between what we do internally as a company and then the industry. So Lee worked as a commercial director of Cell and Gene Therapy Catapult. It's a really large research and manufacturing organization, government funded, and he's worked across 100 plus startups in the industry across the world, and he has that kind of insight and access to industry. So that's crucial actually, because we can think we're doing a good job, but ultimately a good job is what the end users define as a good job and what they need. So we're always talking to them and getting steers from them. And so through those interactions, we've started a couple of small scale paid partnerships with some smaller therapy developers. But as we move forward now, we're really just going to be talking to slightly larger organizations and pharma and trying to look at taking some of their assets and seeing how we can improve them in vivo with our DNA designs.
Quite the transition from academia to venture capital and then to setting up your own company. And they seem to be doing very well, thank you very much. So we wish Houdini Bio all the best as they progress. So that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact us at jim@deeptechdigest.com. So until next time, wherever in the world you are, I hope you have a great week ahead. Stay cool if you're in a heat wave. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.